Heat shock proteins are the cell's emergency repair crew, but they're also its thermostat. When a fever spikes, these tiny machines scramble to refold misfolded proteins, yet they do it by sensing temperature changes, not just fixing damage. How can a protein that is activated by heat act as a cooling system? Stay tuned to see how the cell keeps its balance. After seeing the heat shock proteins in action, let's pause and look at what they actually do. These chaperone proteins bind unfolded polypeptides, shielding them from aggregation. They use ATP to cycle between binding states, guiding the chain into its native fold. This process restores protein homeostasis, keeping the cell functional even when temperatures rise. Heat shock proteins act as cellular thermostats. When temperature rises, proteins begin to unfold. Unfolded chains expose hydrophobic patches that can stick together. The cell's first line of defense is a rapid increase in heat shock protein production. These chaperones bind the exposed regions and shield them from aggregation. While bound, they keep the polypeptide in an extended state ready for refolding. ATP hydrolysis provides the energy to cycle the substrate through folding intermediates. Once a native fold is achieved, the heat shock protein releases the chain. This cycle restores the balance of properly folded proteins inside the cell. Without this mechanism, cellular function would collapse under thermal stress. PAW 0.8's heat shock proteins act as cellular thermostats. They bind unfolded polypeptides to prevent aggregation. Binding exposes hydrophobic patches that would otherwise stick together. The complex is then directed toward refolding or degradation pathways. ATP hydrolysis provides the energy for conformational changes. Chaperone rings encapsulate substrates, creating a protected folding chamber. Release occurs once the protein achieves its native structure. Misfolded proteins are recycled or targeted to proteasomes. This cycle maintains intracellular protein homeostasis during stress. Thus, heat shock proteins restore folding balance and cell viability. PAW 0.8's when a cell experiences heat or other stress, proteins begin to unfold. Unfolded polypeptides expose hydrophobic patches that attract each other. These exposed regions promote aggregation, creating a visible load of misfolded proteins. The cell senses this accumulation through a surveillance network that monitors protein quality. A key sensor is the transcription factor heat shock factor 1, or HSF1. In unstressed conditions, HSF1 remains monomeric and inactive in the cytoplasm. When misfolded proteins accumulate, HSF1 binds them indirectly through co-chaperones. This binding releases HSF1 from inhibition and triggers its trimerization. The active trimers then translocate to the nucleus, where they bind heat shock elements on DNA. Binding initiates transcription of genes encoding heat shock proteins that will later refold or degrade misfolded substrates. Thus, the first causal step is the detection and signaling of protein misfolding by HSF1. This event sets the entire heat shock response in motion, preparing the cell to restore folding balance. PAW 0.8's after the initial unfolding, heat shock proteins bind to exposed hydrophobic patches. This prevents aggregation by shielding reactive residues from each other. The binding event triggers a conformational change in the chaperone complex. ATP hydrolysis supplies energy for this structural rearrangement. Once bound, the substrate is positioned within a protective cavity. The cavity mimics a native-like environment that favors correct folding. During refolding, the chaperone cycles through open and closed states. Each cycle allows the protein to sample multiple conformations. Successful folding releases the substrate back into the cytosol. The heat shock protein then resets for another round of assistance. PAW 0.8's finally, the chaperone cycle culminates in the release of the refolded protein back into the cytosol. The ATP-bound HSP70 disengages from the polypeptide once the folding intermediates reach a native conformation. Cofactor HSP40 had earlier delivered the substrate and guided it toward the active site of HSP70. During this handoff, the hydrophobic patches that were exposed during stress are now buried within the protein core. The restored native structure is then free to participate in its physiological role without aggregation. Because the chaperone cycle consumes ATP, it acts as a molecular thermostat that adjusts folding capacity to heat load. When temperatures rise, more HSP-70 molecules are synthesized and recruited, amplifying the refolding throughput. Conversely, at normal temperatures, the system downregulates, conserving energy while maintaining protein homeostasis. Thus, the final causal step is the ATP-driven release of correctly folded proteins, which restores intracellular folding balance. This balanced state ensures cellular functions continue smoothly even after thermal perturbation. Pause 0.8 Heat shock proteins act as cellular thermostats, regulating protein folding. When temperature rises, misfolded proteins accumulate and threaten cell viability. Chaperones bind exposed hydrophobic patches to prevent aggregation. Binding is transient. ATP hydrolysis drives release and refolding cycles. Strong performance requires sufficient chaperone concentration and rapid ATP turnover. Weak conditions arise when ATP supply drops or chaperones are depleted. Under weak conditions, misfolded proteins persist, forming insoluble aggregates. Aggregates sequester functional proteins, reducing overall folding fidelity. The cell compensates by upregulating heat shock protein synthesis. However, if the stress is prolonged, compensation fails and damage accumulates. Pause 0.8 Heat shock proteins function as cellular thermostats during temperature stress. They bind misfolded polypeptides to prevent aggregation. ATP hydrolysis drives the chaperone cycle of binding and release. Released substrates then refold into their native conformations. Unsuccessful folding leads to proteasomal degradation. Maintain protein quality. This dynamic balance preserves cellular homeostasis under heat shock. In industrial bioreactors, engineered HSPs enhance enzyme stability at high temperatures. Their presence reduces product loss and increases overall yield. Similarly, crop plants upregulate HSPs to survive drought and heat. Transcription factors such as HSF1 trigger this protective expression program. Thus, molecular thermostats exemplify a universal strategy for proteostasis. Understanding their mechanism informs design of resilient bioprocesses and agriculture. Pause 0.8 Heat shock proteins act as cellular thermostats during stress. When temperature rises, protein folding becomes unstable. Misfolded proteins accumulate and threaten cell viability. HSPs bind to exposed hydrophobic patches on these proteins. Binding shields the misfolded regions from aggregation. The complex is then directed to refolding machinery. ATP hydrolysis powers the remodeling of the protein structure. Once correctly folded, proteins re-enter normal cellular pathways. If refolding fails, HSPs target them for degradation. This cycle restores intracellular folding balance after heat shock. The expression of HSP genes is upregulated by heat shock factor 1. This transcriptional response ensures a rapid increase in available chaperones. Pause 0.8 Heat shock proteins are activated when cellular temperatures rise above normal. The stress signal triggers a transcriptional cascade that increases HSP gene expression. Elevated HSP levels bind to exposed hydrophobic patches on unfolded polypeptides. This binding shields aggregation-prone regions and prevents misfolded protein clumping. Simultaneously, the chaperones reposition the polypeptide into a folding-competent conformation. ATP hydrolysis supplies the energy needed for conformational rearrangements within the HSP complex. Once properly folded, the protein is released and can resume its functional role. If refolding fails, the HSP system targets the misfolded species for proteolytic degradation. Thus, heat shock proteins act as molecular thermostats that restore intracellular folding equilibrium. The cycle ends when temperature normalizes and chaperone synthesis returns to baseline levels. Pause 0.8 Heat shock proteins detect misfolded proteins, bind them with ATP-driven cycles, refold them into functional structures, and thereby maintain cellular protein balance during heat stress.